This is the Professor, CBMD PhD. Today's Friday, November 15th, 2024. It is 10:22 and 14 seconds, p.m. Eastern Standard Time. And today's research, Microdomains. So let's hop to it here. The fluidity of the plasma membrane is not revealed in static electron micrographs. Experiments reveal that the membrane behaves as though it were a two-dimensional lipid fluid. For many years, it was thought that integral membrane proteins move freely within the plane of the membrane. This movement was compared to the movement of icebergs floating in the ocean. However, the distribution and movement of proteins within the lipid biolayer is not as random as once thought. The plasma membrane appears to be patchy, with localized regions that were distinct in structure and function, and vary in thickness and composition. These localized regions contain high concentrations of cholesterol and glycosphingolipids and are called lipid rafts because of the high concentration of cholesterol and the presence of longer, highly saturated fatty acid chains. The lipid raft area is thicker and exhibits less fluidity than the surrounding plasma membrane. Cholesterol is a dynamic glue that holds the raft together. Its removal from the raft results in dispersal of raft-associated lipids and proteins. In general, there are two types of lipid rafts. Planar lipid rafts contain a family of 47 kDa proteins, known as flotillins, as well as specific lipids and cholesterol. Flotillins are regarded as a molecular marker of the lipid rafts and are considered to be scaffolding proteins. They also participate in the recruitment of specific membrane proteins into the rafts and work as active partners in various signaling pathways. Cavular rafts or cavola. Little caves represent small 50 to 100 nanometers in diameter. Flash-shaped investigations of the plasma membrane containing 18 to 24 kDa integral membrane proteins called cavolins. Oligomerization of cavolins produces a cavolin scaffold essential to creating investigations within the plasma membrane. These proteins also bind to cholesterol and a variety of proteins involved in signal transduction. Caveoli are prominent smooth muscle cells where they contain various cadmium channels, sodium-cadmium exchangers, and G-protein, coupled receptors involved in ligand-mediated regulation of intercellular cadmium level. Lipid rafts contain a variety of integral and peripheral membrane proteins involved in cell signaling. They can be viewed as signaling platforms floating in the ocean of lipids. Each individual raft is equipped with all the necessary elements, receptors, coupling factors, effector enzyme, and substrates to receive and convey specific signals. Signal transduction in lipid rafts occur more rapidly and efficiently because of the close proximity of interacting proteins. In addition, different signaling rafts allow for the separation of specific signaling molecules from each other. And this is Professor Christopher, codename Will, leader of research scientist ufologist, computer technician, computer programmer, philanthropist, writer, mathematician, artist, photographer, film producer, chef, animal, and nature lover, passion, world peace. And this video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day. This is the Professor, CBMD PhD. Today's Friday, November 15th, 2024.